Yeah, Lukas and I are working for DataPlant, which is part of the NFDI, which yeah, it's like from the German Research Foundation. Yeah, we're working on research data management for very different fields, and we have like the plant biology project. Uh, we're developing the ARC. Um, uh, as like a metadata or research data management framework. Um, and we are at the moment closely tying our formats to RO-Crate and using schema.org, and that's why we are that's how we got um, into here into bioschemas. And today I want to talk about the current work of the LabProtocol group, which means the types LabProtocol and LabProcess. And I think this work has roughly been started 2 years ago at the biohackathon, uh, where we basically restructured the LabProtocol working group. As far as I remember, it was not a new group, but yeah, we kind of took over the work on those types. So um, uh, what we are trying to model here are like all kinds of laboratory processes where you have some entity, a cell, a plant, uh, a chemical, whatever, and then you have processes that connect them. So you take one sample, produce a new sample, you take a sample, do a measurement, um, you get some data, and we want the generic framework for all these laboratory processes. Um, and then we want them in an annotated way. So the idea is we have these two kinds of objects, entities or materials, and then processes or actions between them. And both should be annotated with parameters, characteristics, whatsoever. And the approach that we're taking here is like we want a very generic way to annotate them with terms from any biological or technical ontology, um, which we want to achieve in a very similar way to the already existing type sample. So the uh, I mean, there's also work going on with sample at the moment, but the previous version of sample basically was just a thing with additional properties, and these additional properties are property values, which can then be annotated as any key value pair. And we want the same thing for the processes that connect these samples. That's the overall strategy or the annotation syntax that we're aiming for. And then in addition to this, we want to incorporate basically this perspective that's also present for computational workflows, the separation between prospective provenance and retrospective provenance. So if we look at the RO-Crate work or the workflow run crate profiles for RO-Crate, then there you have basically computational workflows which describe kind of programs or tools or whatever, and then the executions, the runs of those workflows, programs, whatever, um, where the type for the runs is a schema.org create action or just an action that then has input and output properties pointing to objects files which are inputs which are consumed and produced by a computational workflow. And we want the same thing for real world processes uh, in labs. Um, so we have like these if you look at the hierarchy there uh, for computational workflows, we want this here on the right. We want the same thing for laboratory processes. So you have like one data set. This has a list of lab processes, and they are connected to lab protocols, which describe like the prospective perspective of on those processes. And actually, in DataPlant or in the ARC, we use these types already in several places. So we use them to model the ISA part of or the experimental part of the arc, which is like an adaptation of the ISA model. And then we also use those same types in conjunction with the workflow run crate types and profiles for the computational workflows in an arc. So this is already like established in our tooling. And so why do we need changes in bioschemas for this? Well, there was this LabProtocol type, but the old LabProtocol type was very ambiguous about the separation. So it kind of like was both a process, as I described it before, and a protocol. So like, for example, in the description was not very clear. It said an experimental protocol is a sequence of tasks and operations executed to perform experimental research, which doesn't make it clear, is it like the prospective description or the retrospective description? And also the properties or attributes assigned to it were not very clear. So for example, you had a purpose and lab equipment, which both like, yeah, are very generic and can be assigned to a protocol in the prospective sense. But then also you have that a specific sample used or an execution time, which is like a property of the actual execution of this protocol. And the solution that we thought of was like, yeah, separating this, having a LabProcess type and a LabProtocol type. And now the description also make it clear that the LabProcess is the retrospective description and the protocol is the prospective description. And the LabProcess type is designed in analogy with the sample I said before. So we have this property parameter value that just points to a property value, and this is just a key value pair where you can assign anything that you want. You can have like a temperature as the key, and then the specific temperature value, 35 degrees as the value. And then you also have the executes LabProtocol, which then points to the protocol that has been used for this process. And then for the profiles, we reduced the actual requirements significantly. So previously, the LabProtocol profile had as a minimum requirement a biosample intended use keywords, lots more. And in the revised version of the profile, we basically, as a minimum, we just have a name. And then all other properties are either recommended or optional. And the same holds for the LabProcess. So the LabProcess profile is also, yeah, it has very little hard requirements for it to be valid. And then lastly, I want to talk about like where we already use this. So I said our tooling makes a lot of use of these types already. And we have for RO crates, we have the ISA RO crate profile, which is also developed by DataPlant. Here we use, we make use of LabProcess and LabProtocol as the core of all these profiles. So here we have like the overall scheme of like or diagram representing like the dataset as a whole. And as you can see, LabProtocol and LabProcess are core ingredients together with the bioschema sample. And basically the three main types in how we annotate datasets. And then these RO crates are already used in the data hub for data publications as ARCs, which you can see here for I don't know about like this publication in detail. Maybe Lukas knows more about it. But yeah, they are in active use already, although there are still drafts in bioschemas. So this is for now all I have to say, and I'm happy to discuss anything about it or answer any questions in the following.